Hello everybody, my name is Aastha Guragain and I am pursuing a master's degree in bioinformatics at Grand Valley State University. Today we are excited to present our work on interactive visualization of biological concepts using natural language processing in a graph database. This project bridges literature mining and network biology to make biomedical research more intuitive. Let me introduce my colleague Mahendra who will walk you through the technical details.
Thank you, Aastha. Hello, everyone. I'm Mahendra Sarata. I'm from the wing of Data Science and Analytics from Grand Valley State University. Coming to this research, our goal was to tackle a major challenge. Biomedical literature is vast and complex, but traditional tools are text-heavy and hard to navigate. So we built a system that transforms PubMed abstract into interactive graphs, connecting genes, diseases, and pathways, while adding a chatbot for natural language queries and a graph database for graph queries.
Talking about the approach and architecture, here is how it works. We start with a mesh-based PubMed queries to fetch abstract. Then tools like PubData extract the entities such as genes, pathways, species, and gProfiler maps them to the pathways. But the real innovation is in the architecture.
Exactly. We combine LLMs like Olama and GPT to power a literature chatbot and also generate summaries for the graphs. Abstracts are processed, stored in PyStore DB for caching, and relationships are visualizing using D3.js graphs backed by Azure Cosmos DB with Apache Gremlin support. Users can query in two ways, natural language or technical graph queries, thanks to the LLM translator.
Exactly. So, what makes this application unique? First, the dual query interface. Researchers can use precise graph queries using Gremlin API, while clinicians or students can ask questions in plain English. Second, the chatbot pulls answers directly from embedded abstracts using semantic search and also generates summaries for the updated visualizations.
Like, for example, searching genes related to two-bone tissue cancer or any other cancer out there, which exactly generates an interactive graph and highlighting pathways and co-occurring entities. It's more of like a Google Maps for biomedical relationships.
Talking about the results and the accuracy of the web application, our application is 100% accurate in obtaining articles from PubMed. Along with that, it displays the species, diseases, chemicals, and pathways connected with specific PMIDs in the form of nodes and edges. Further, the genes obtained by the web application were enriched using gProfiler for the rat species, and it was identified that 80% of the enriched pathways were retrieved by the web application correctly. But we are just scratching the surface here. Next step include full text article integration and personalized user profile to save the queries.
And along with that, we also plan to improve summaries graphs and visual filters for diseases or species, image dragging a slider to see how gene disease interacts links change over time. Ultimately, this tool could accelerating track repurposing and collaborative research.
So to recap, we merged natural language processing, graph database, and large language model to turn static literature into dynamic, explorable knowledge.
Please go ahead, use our link to understand more about our research and use our tool. Thank you so much.
Thank you.